Can somebody confirm that you can hear me?
Yes.
Thank you. I apologize for being late. I've been having trouble logging in. Before we begin, are there any questions? I think we'd finished chapter 4. All right, if there's no questions, let's begin. I'll share my screen. All right, you should be able to see my screen now. Today is October 24th, and we should be on chapter 5. There is a lab today, lab 6. Any questions about any of that? If not, let's move on to chapter 5. Chapter 5 is microbial metabolism. As always, I show you an introductory slide listing the major goals and a rough outline. Know the terms of this chapter. We'll talk about catabolism, anabolism, activation, energy, an enzyme. What's an active site of an enzyme? What's competitive inhibition, non-competitive or allosteric inhibition? What's a redox reaction? What are photoautotrophs, photoheterotrophs, chemoautotrophs, and chemoheterotrophs? What is fermentation? And then lastly, we'll talk about cyclic and non-cyclic photosynthesis. So that was number 1. Number 2, understand what feedback inhibition is. And 3, understand what aerobic respiration is, its steps, and how it differs from anaerobic respiration. Any question about any of that? All right. Microbial metabolism is all of the chemical reactions within a cell. And for a microbe, because the microorganism is a cell, this is also all of the chemical reactions within the organism. When we have metabolism, we can break it down into two parts, catabolism and anabolism. Catabolism is a decomposition reaction where you break down a larger molecule, such as starch, into its smaller subunits, such as glucose. Energy is given off by this reaction because a larger molecule stores more energy than the small molecules. Like starch has more energy than all of the glucose parts. Some of that energy is given off as heat. Whenever you transfer energy from one form to another, heat is given off. It's the second law of thermodynamics. Some of that energy can be used to make ATP. However, this slide is oversimplified because in animal cells, like our cells, we only have two chemical processes or two chemical reactions that can make ATP, and that's aerobic respiration and fermentation. We'll talk about both of those in this lesson. All of the other catabolic reactions do not generate ATP. Any question about any of that? So even green plants, which are much better at making ATP than animal cells, most of their catabolic reactions cannot be used to make ATP. Anabolism is a biosynthetic reaction where you take a smaller molecule, such as glucose, and then hook them all together to make a bigger molecule, such as starch. You need energy to enter the system because starch has more energy than glucose. And so this is an energy using reaction. Most of the time, that energy comes from ATP. we break down ATP and then use that energy to make the anabolic reaction. Now, some of that energy will be lost as heat. And that's because, like I said, the second law of thermodynamics, whenever you have energy in one form, such as the energy in glucose and the energy given up by ATP, and transfer it to another form, such as starch, energy is given off as heat. Let me state that heat is just another form of energy. It is a form of energy that living cells cannot readily use. The exception is a warm-blooded creature can use some of that energy to keep the temperature of the creature higher. Of course, in humans, if we generate too much heat, then we have to uh, sweat to give off that heat. Any question about any of that? All right. A metabolic pathway is a sequence of enzymatically catalyzed chemical reactions in a cell, such as this metabolic pathway. Let me see if I can blow that up. We have molecule A, which is a starting substrate, and then we have an enzyme, which makes molecule B. Molecule B is the product of this first reaction. However, molecule B is the starting substrate of the reaction 2, which enzyme 2 makes, making molecule B molecule C. Molecule C can be in turn made by enzyme 3 into molecule D. Molecule D can be in turn made into molecule E by enzyme 4. And then molecule E can be made by enzyme 5 into molecule F. Now, molecule F is the last product that's made of this chemical reaction, so we call that the end product. So A is the starting substrate, also called the starting reactant, and then molecule F is called the end product. All of these other molecules are called intermediate or intermediate products because they are the product of the first reaction, but then it's the start of the next reaction. Any question about any of that? All right, metabolic pathways are determined by enzymes, and enzymes are encoded by genes. So metabolic reactions are determined by an individual's genes. Here is a huge amount of metabolism being shown in this slide, and we'll talk about all of it. And of course, I'll quiz you on all of it. Any questions? Only joking. We won't talk about all of that chemical reaction going on in the cell. And this slide is also, oh gosh, probably 15, 20 years old, so it's also probably out of date. Chemical reactions occur by the collision theory. The collision theory states that chemical reactions may occur when atoms, ions, or molecules collide. All they have to do is have enough energy in the colliding atoms, ions, or molecules, and then you have the reaction occurring. Activation energy is the energy needed to disrupt the electronic configurations in the starting atoms, ions, or molecules to make the products of the chemical reaction. The reaction rate is the frequency of collisions with enough energy to bring about a reaction. You can increase the reaction rate by increasing the abundance of enzymes. If you have more enzymes, an enzyme is more likely to get together with the starting substrates and then speed up the chemical reaction. You can also increase the reaction rate by increasing the temperature If you increase the temperature, what you're doing is you're increasing the movement of the atoms, ions, and or molecules so that they're more likely to collide, and they're more likely to collide with more energy, increasing the reaction rate. Now, pressure is similar. What you're doing is you're putting the atoms, ions, or molecules under pressure so that when they collide, they have more energy and you're more likely to speed up the chemical reaction. Any question about any of that? This graph is showing you the activation energy of a chemical reaction without an enzyme and then with an enzyme. So on the y-axis, we actually have the energy level. And as you can see, the starting reactant or starting substrate has more energy than the products. That's because the reactant is a larger molecule. And we can think of this as something like sucrose. Now, let's go glucose. And it has more energy in the chemical reactions of the molecule than the products, which in this case would be CO2 and water. Glucose can be broken down into CO2 and water. Now, for glucose to change into CO2 and water, energy will be given off because the products have less energy. But to move from this position to that position without an enzyme, the reactants have to get energy, and they have to get what we call the activation energy without an enzyme, which is an energy hump, to make the products. And that's why if you had a cup of glucose and you looked at it a day later, or even a year later, you'll still have a cup of glucose. Every once in a while, a molecule of glucose will get enough energy that it'll break down to CO2 and water, but that won't happen very often. And even a year later, you'll still have essentially a cup of glucose. I can get this glucose to automatically turn into CO2 and water, and that is if I took a blowtorch to the glucose, I would burn it and convert it into CO2 and water. And that tells you how much energy you need to add to glucose to get it into CO2 and water. You have to get the temperature of a blowtorch, which I don't remember if it's 1,000 degrees Fahrenheit or 1,000 degrees Celsius, but it's an awful high temperature. Now, to get glucose to change into CO2 and water, it happens all the time in the room you're in. Where does it happen in the room you're in? Come on, somebody's got to know. Does it happen in the air you're breathing? Assuming you're not breathing in microbes. Does it happen on the table? Catching the table on fire? Anyone know where that happens? You guys have to contribute. It happens in your cells. Glucose all the time is broken down from glucose into CO2 and water. Let me see how many people are here. There's 7 of you. And obviously, the temperature of your cells is much less than 1,000 degrees. That's either Fahrenheit or Celsius, I don't remember which. And so the reaction rate with an enzyme does have an energy hump, but that energy hump is much less than the energy hump without the enzyme. And so this is not really drawn to scale because this is only a slight increase I don't know, 25 to 37 degrees Celsius. And this would be something like 25 to 1,000 degrees Celsius, or maybe it's like 800 degrees Celsius. I don't remember, but it's either 1,000 degrees Fahrenheit or 1,000 degrees Celsius. It's a big energy difference. And this is called the activation energy with the enzyme. That's the activation energy without the enzyme. Any questions about any of that? Enzymes are biological catalysts. A catalyst is something that speeds up a chemical reaction. They can speed up a chemical reaction 10 to the 8th to 10 to the 10th times. Enzymes are encoded by genes. Enzymes are also specific for a single chemical reaction. This is a bit of an oversimplification. Those of you who know about some of the drugs we do have some drugs that will work on a enzyme, and what they're doing is they work competitively with the substrate to bind to the enzyme. But generally speaking, enzymes are very specific, and we're going to oversimplify it and say one enzyme works on one molecule. Like I said, it's not quite that specific, but it's close to it. We have a different enzyme for breaking down sucrose, a different enzyme for breaking down maltose, a different enzyme for breaking down lactose. So that in this sense, uh, an enzyme is specific for one single chemical reaction. The specificity is due to the enzyme's three-dimensional shape. The three-dimensional shape allows the enzyme to bind to one and only one, generally, specific substrate. What exactly binds to the substrate is the enzyme's active site. And it's the three-dimensional shape of the enzyme creating this active site, which can bind to one single substrate. Enzymes are not used up by the chemical reaction. So once an enzyme binds to a substrate, it can then change the substrate into the product, release the product, and then the enzyme can bind to another substrate. Any question about any of that? So an enzyme can be used time and time again. The form of the enzyme substrate complex allows the substrate to orient so that it increases the probability of the chemical reaction, meaning it puts stress on the chemical bonds of the substrate to help change the chemical bonds of the substrate into the chemical bonds of the product. You can think of it as a big molecule. You're putting stress on one of the chemical bonds, and then that chemical bond will break, creating two smaller molecules that we call the products. And it's a little more tricky to think of it in the other word, and that's where you have two smaller molecules joining to form a product. But once again, the chemical bonds are oriented such to switch the chemical bonds of the substrate into the chemical bonds of the product. It is this enzyme substrate complex that is lowering the activation energy. Any question about any of that? Let me get out of my mail here. I have to get into my mail to log in, and so that's why I have the mail there. There we go. Now, where was I? Here we go. Enzymes do have a classification naming system, and that is all enzymes normally end with ASE or ACE, like catalase, lyase, isomerase, ligase. We'll talk a little bit about catalase, although I already talked about it in the lab, and then ligase. All of these others you don't need to know. These are just examples of enzymes. Not all enzymes end in ase, particularly any enzyme that was named before this naming convention started, like the enzyme renin, which curdles milk in a baby's stomach so that the milk stays in the stomach longer to be digested better. Renin is an enzyme that doesn't end in ase, but normally enzymes do end in ase. You should also know that enzymes don't work in a vacuum. Some enzymes require a helper. That helper is either a coenzyme, that's a small biological molecule, or a cofactor. This is usually a ion like iron, zinc, magnesium. And the coenzyme binds to the enzyme to make the complete enzyme, which then will have the three-dimensional shape to bind to the substrate. You should know a coenzyme is a biological molecule. We'll talk about a few of them. And then a cofactor is just something smaller. Generally, it's an ion of an element, which is usually a metal element, such as iron, zinc, magnesium. You do not need to know the name of the enzyme before the coenzyme and the name of the enzyme after the coenzyme binds to it. So I'm not going to test you on any of the names except obviously a coenzyme and a cofactor. You should know those. Here is listing some important coenzymes. NAD plus, which probably means as much to you as nicotinamide adenine dinucleotide. So you can call it either way. Generally in this class, we'll call it NAD plus. It's a niacin derivative, primarily involved in catabolic reactions. We will talk about NAD plus in aerobic respiration. So you should know that NAD plus is a coenzyme. FAD, flavine adenine dinucleotide, is also a coenzyme that we're going to talk about in this lesson because it's important in aerobic respiration. So you should know these first two coenzymes. The others we're not going to talk about, although I guess we will talk a little bit about coenzyme A. So you really don't need to know the others, but you should know NAD plus and FAD are important coenzymes, and we will talk about them in aerobic respiration. NADP plus is actually found in photosynthesis, and we're not going to talk much about photosynthesis. It works similar to NAD plus. Any question about any of that? All right, let's get back to how an enzyme works. I mentioned that an enzyme can bind to a substrate on its active site, and the active site has a three-dimensional shape that specifically binds to a substrate. So this is showing you the lock and key model of how an enzyme works. The enzyme works as a key to unlock the lock or the substrate, binding specifically to it, putting stress on the chemical bonds of the substrate to make the chemical bonds of the product. The product then falls out of the enzyme, and then that enzyme can go and rebind to another substrate. That is the lock and the key model of how an enzyme works. This graph is showing you the hand in the glove model of how an enzyme works. And there's three things you can identify from this picture. Although one of them, I've got to blow this up or else you won't notice it. What do you notice about the hand in the glove model of how an enzyme works? Two of them are very obvious. Is anyone going to contribute?
Does the enzyme just catches the enzyme, right? Or like
Yeah, the
uh,
active site right here catches the substrate, you mean?
substrate, yeah.
Yeah. What do you notice about the enzyme? Before it touches the substrate, and then after it's in a complex with the substrate.
It looks like it's like open and then it
Yeah,
closes itself around it.
yeah, that's why we call it the hand in the glove model. And that is an enzyme acts like a glove and then molds to the substrate, like the glove molds to your hand. Another difference is very obvious that the enzyme is much larger than the substrate. And that we actually saw before here. Let me see if I can blow that up again. The third one would be very difficult to notice, but if you look at the substrate here and the substrate there, this substrate is a little flatter than that substrate. I like to call this the shapeshifter's hand going in the glove, where the glove molds to the shapeshifter's hand, but the shapeshifter's hand molds to the glove. Those of you who know about Star Trek Deep Space Nine know what I'm talking about when I say shapeshifter, but the shapeshifter can change the shape of his hand so it'll then mold to the glove. Any question about any of that? Let me shrink this down. Um, I guess I already did that. So an enzyme and a substrate can be said to work as the lock and the key. And in your textbook, they talk about the hand in the glove model. And I extended it further by saying it's also, you can say it's a shapeshifter's hand in the glove model, how an enzyme actually works. And your textbook doesn't talk about the shapeshifter's hand in the glove model. Enzymes do not work in a vacuum. I've already stated that. Enzymes do have factors which can influence the enzyme's activity. Like an enzyme has to work in the correct temperature, the correct pH, the correct salinity. So temperature, pH, and salinity are three factors that can affect an enzyme's activity. Heavy metals can also, generally heavy metals inactivate an enzyme. Alcohol inactivates an enzyme. UV radiation can also inactivate an enzyme. All of these factors can cause the protein to denature. By denaturation, we change the enzyme shape, the protein shape, so that it loses its three-dimensional shape. Yeah, I don't know how to word that. Let me go over here. A three-dimensional active enzyme, one that's lost its shape and has become denatured. And you can start to lose the enzyme's three-dimensional shape by having the enzyme at the wrong temperature, the wrong pH, the wrong salinity, the presence of heavy metals, the presence of alcohol, and the presence of UV radiation, causing the protein to denature. Another factor that can influence the enzyme's activity is the substrate concentration. Like if the enzyme is around, but there's no substrate, obviously there's no enzyme activity. And as you increase the substrate concentration, you can increase the speed that the enzyme works at, meaning increasing the enzyme's activity. Poisons can also affect the enzyme's activity. Generally, poisons will decrease the enzyme activity, like cyanide and fluoride. These are two poisons which actually inactivate enzymes. And if you have enough of these enzyme poisons around, the enzymes won't function, and of course that will then poison you and then kill you. Any question about any of that? All right. We can also influence the enzyme's activity by inhibition. We can have two types of inhibition, competitive inhibition and non-competitive or allosteric inhibition. And that's where something can bind to the enzyme and then inactivate the enzyme, inhibit it. And then lastly, we'll talk about feedback inhibition, where you can have feedback inhibition controlling an enzymatic pathway so the cell only makes so much of one product. So I've already talked about this side, and that is enzymes can be denatured. Full denaturation is where the active functional protein loses its shape, loses its folding pattern, and now the enzyme cannot work at all. Fully denatured. You don't have to be fully denatured. You could just be partially denatured, where the enzyme slightly has a change in its shape, and then it doesn't work as well, but it still can work. So with temperature, this enzyme works best at about 37 degrees. So that's probably one of our enzymes. And as you decrease the temperature, you partially denature the enzyme, meaning it's starting to unravel, either decrease or increase the temperature. The enzyme's still able to work, but at a less rate than the optimal rate. And then you decrease the temperature further or increase the temperature further, the enzyme loses more activity, becomes more denatured, but not fully denatured. And then you decrease the temperature dramatically or increase it dramatically, and now you have the enzyme totally denatured. The enzyme cannot function. This one less than 10 degrees or more than 50 degrees Celsius. And at this temperature, or that temperature, we say the enzyme is totally denatured. Any question about any of that? The same thing happens with pH. With this enzyme, its optimal pH is happening around pH 5. Does anyone know where an enzyme in your body works at an optimal rate around pH 5? I'll give you a hint. It's not the stomach. The stomach's between pH, I don't know, 2 and 1.5.
The
The
esophagus? That's a total random guess.
I'm not sure the pH of your esophagus. The pH of your mouth is acidic. I'm not sure what the pH of even your mouth is, so it could be. I was referring to the pH of your skin. That one is one I know is around pH 5. So, like I said, the esophagus could be around pH 5. For one thing, you breathe in CO2 and it will change the water in your mucous membranes to somewhat acidic. And it might be pH 5. I don't know. So you could be right. The point is that when you change the pH a little bit, you decrease the enzyme's activity. The enzyme is partially denatured, and then you can denature it further, but the enzyme still has activity. And this enzyme can go to pH 0 before it becomes totally denatured, has no activity, or pH 10, like you're at pH 11. You have no enzyme activity from this enzyme. Once again, enzymes do not work in a vacuum. They have to work at their optimally, they have to work at their optimal temperature and their optimal pH. The substrate concentration level can also affect an enzyme's activity. Obviously, when you have no substrate, 0, there is no enzyme activity, and you increase the substrate concentration, you dramatically increase the enzyme's activity rate. And that's because now the enzyme can bump into the substrate and then convert it into the product. But as you notice, you increase the substrate concentration further, the reaction rate starts to slow. And eventually, when you increase the substrate concentration further beyond about this point here, you do not get any increase in the amount of the enzyme's activity. The reason why there is no increase in the enzyme activity once the substrate is this high or higher is because at this level, all of the enzyme is currently already bound to a substrate. And if you increase the substrate concentration further, you cannot increase the enzyme's activity rate any higher because all of the enzyme is already bound to a substrate. You can't get higher than 100%. Any question about any of that? All right, I mentioned that uh, there is another factor which can influence an enzyme's activity, and that is inhibition. An inhibitor can bind to an enzyme and then inhibit the enzyme's ability to function. Competitive inhibition, in a competitive inhibitor is an inhibitor that binds to the active site of the enzyme. And it's called a competitive inhibitor because the inhibitor is competing with the substrate for the active site. And we have some drugs which are competitive inhibitor, and what you do is you give a high dose of the drug, and then the drug is more likely to bind to the enzyme and then shut down this chemical reaction. Any question about competitive inhibitor? When the competitive inhibitor is bound to the enzyme, then the product cannot bind. A non-competitive inhibitor or an allosteric inhibitor, these two terms are used interchangeably. They interact with the site other than the active site on the enzyme. So here the non-competitive inhibitor binds to the enzyme, and that changes the three-dimensional shape and folding pattern of the enzyme such that the active site is changed and no longer can bind to the substrate. And that's how a non-competitive inhibitor inhibits the enzyme. This other site that the non-competitive inhibitor binds to is called the allosteric site. Any question about any of that? All right, our last factor for influencing an enzyme's activity is feedback inhibition. This is a type of non-competitive allosteric inhibition. In feedback inhibition, you have an enzymatic pathway. So here we have the first reaction, changing the substrate into intermediate A, and that's done by enzyme 1. You also have enzyme 2, which makes intermediate A into intermediate B. And then we have enzyme 3, which converts intermediate B into the end product. In feedback inhibition, the end product itself can bind to enzyme 1 and act as an allosteric inhibitor, changing the binding and allosteric, not the allosteric, the active site of the enzyme. So the enzyme can no longer bind to the product, shutting down the enzyme. In this way, a cell, if it uses feedback inhibition, can maintain the level of end product in the cell. And that is when the end product is low, there will be no enzyme binding to enzyme 1, and then the enzymatic pathway will occur, making the end product. As the end product climbs in concentration in the cell, more and more of the end product will bind to enzyme 1, starting to shut down enzyme 1 until you get the end product at a high level, in which case it's totally bound to enzyme 1 and then you shut down the production of the end product. And that means you'll get no more production of the end product. And so the level will never increase beyond what it's at currently. But as the cell uses up the end product, there will be less and less end product around. And every once in a while, the end product will fall off. And then that enzyme 1 will begin to work. And as the end product concentration lowers in the cell, you'll have less and less end product binding to enzyme 1 until all of enzyme 1 is working to make more end product. And like I said, this allows the cell to keep the concentration of the end product at a lower limit, meaning below that level, the enzyme will be turned on and it'll make lots of end product. And then the upper limit, I don't know if you can see my hands here or not, the upper limit is where there's enough end product around that it totally shuts down the chemical reaction. So feedback inhibition is a way for the cell to maintain the end product at a certain level within the cell, certain level of concentration within the cell. And we normally find a feedback inhibition like this, where the end product itself binds to the enzyme 1, doesn't bind to enzyme 2, because if it did bind to enzyme 2, you do shut down the chemical reaction, but you're spending energy making intermediate A. And a cell wants to be energy efficient, and the most energy efficient is to have the end product itself shut down enzyme 1. And that's why feedback inhibition is normally seen like this, as shown in this slide here, where the end product binds to enzyme 1. Any question about any of that? All right. I have at times stated that an enzyme is like a protein, and the protein can denature and unfold. Well, not all enzymes are proteins. Some enzymes are actually RNA enzymes. A ribozyme is an RNA enzyme. Most enzymes are proteins. And we're talking about probably over 95% of enzymes are proteins. But some enzymes are RNA molecules, such as an RNA that cuts and splices another RNA molecule, or it could even cut and splice another molecule. RNA enzymes do function as enzymes in that they have an active site that actually binds to the substrate. And then the RNA enzymes are not used up or modified in a chemical reaction. Once the RNA enzyme has bound to its substrate and then changes the substrate into the product, and the product falls off, that RNA enzyme can then bind to another substrate. So an RNA enzyme essentially acts the same as a protein enzyme. The only difference is what the enzyme is made of. There's only one RNA enzyme that I'm going to talk about, and for some reason your book doesn't mention it, and that is we're going to talk about when we're talking about protein translation, that the amino acid being removed from the first messenger RNA will be bound to the growing polypeptide chain that will eventually become the protein. When that amino acid does that, that is happening by an RNA enzyme. So it's the ribosome that does that, but it's the RNA portion of the ribosome. And that's the only RNA enzyme I'm going to mention in this class. Is that true? Well, we do talk about RNA splicing, but I don't mention it as an enzyme. It's actually an RNA enzyme that I don't talk about. Any question about any of that? All right. Let's move on to metabolic diversity among organisms. We're going to talk about microbes, but you should come to realize that this applies to all organisms. All organisms require the following sources. They require an energy source and they require a carbon source. We can categorize the metabolic diversity among organisms based on their energy source. An organism can be classified as a phototroph if they derive their energy from light. Phototroph. We also call it a chemotroph if the organism derives its energy from a chemical compound. We, people, are chemotrophs because we derive much of our energy needs from glucose and other food substances that we consume. Now, we derive our chemical energy needs from an organic chemical compound such as glucose. However, there are some chemotrophs, particularly bacteria and archaea, that can derive their energy from an inorganic chemical compound, such as iron or sulfate or hydrogen sulfide. And these two are chemotrophs. We can also categorize an organism based upon where they get their carbon source. If the organism gets its carbon from CO2, we call it an autotroph. I think everybody knows the green plants get their carbon from CO2, and so they are an autotroph. And then people get their carbon from glucose, and so people are heterotrophs. Well, let me tell you that bacteria and archaea also have autotrophs and heterotrophs. So yes, there is some bacteria which can get CO2, carbon from CO2, and they are autotrophs. And there's more bacteria that are getting their carbon from an organic chemical such as glucose, like E. coli, and E. coli is a heterotroph. If you ever get confused on the trophs, just break it down. Is it a phototroph or a chemotroph? Is it an autotroph or a heterotroph? You can combine all of these sources, the energy source and the carbon source, to get the four trophs defined for organism: photoautotroph, a photoheterotroph, a chemoautotroph, and a chemoheterotroph. For the photoautotrophs, I think everybody knows the green plants are a photoautotroph. They get their light source, their energy needs from light, and they get their carbon needs from CO2. So green plants are a photoautotroph. There's also green bacteria like cyanobacteria, and they get their energy source from light and their carbon source from CO2. We'll talk later about oxygenic and anoxygenic photoautotrophs. An oxygenic photoautotroph makes oxygen, an oxygenic photoautotroph does not make oxygen. And we'll talk a little bit later about that. The next easiest one after the photoautotrophs are the chemoheterotrophs. And we, in animals, are a chemoheterotroph. We get our energy source from glucose, which is an organic chemical compound. And we get our carbon source also largely from glucose, which is a organic chemical compound. Many chemoheterotrophs get their energy source and their carbon source from the same molecule. Like for us, we largely get both sources from glucose. And that's true for many chemoheterotrophs, but not all of them. And we find animals in that, the protozoa, the fungi, many bacteria, like E. coli, is actually a chemoheterotroph. And then the fermentative bacteria, like the bacteria that are in yogurt to change the milk into yogurt. It's a chemoheterotroph. Not all chemoheterotrophs get their energy needs from an organic chemical compound. Some bacteria and archaea can get their energy source from an inorganic chemical compound, such as iron or sulfate or hydrogen sulfide gas. And these are um, I should say, some of the chemosynthetic bacteria getting their energy needs from a chemical compound. They do get their carbon from an organic chemical compound such as glucose. Any question about any of that? All right. Now, I've talked about some of the chemosynthetic bacteria, and that is their chemoheterotrophs, getting their energy needs from an inorganic chemical compound, but they get their carbon from an organic chemical compound, such as glucose. There are some chemosynthetic bacteria, which we call chemoautotrophs, and they too get their energy source from a chemical compound, which in this case will be an inorganic chemical compound, such as iron or sulfate or hydrogen sulfide gas. But unlike the chemosynthetic bacteria, which are chemoheterotrophs, they get their carbon from CO2. So these chemosynthetic bacteria are actually chemoautotrophs. And believe it or not, you actually know a bacteria which is a chemoautotroph. It's an iron oxidizing bacteria, which I'm sure all of you have seen. When you lift up the lid to the toilet tank, you'll see some brownish or yellowish scum, usually on the back wall of the toilet tank. It can be on the sides and the front as well. That is actually iron oxidizing bacteria. Bacteria which is using iron dissolved in the water to get its energy needs. And it's getting its carbon source from CO2. So you all are actually familiar with the chemoautotroph. This is a specific chemosynthetic bacteria, which is a chemoautotroph. And it's on the sidewall of the toilet tank. I'm not talking about the toilet bowl. I'm talking about what stores the water above the toilet bowl, the toilet tank. The fourth group are the photoheterotrophs. They get their energy needs from light, why they're called the phototroph, and they get their carbon needs from an organic chemical compound such as glucose. You're probably not familiar with a photoheterotroph, but the green and the purple non-sulfur bacteria are photoheterotrophs. And I'm sure none of you are familiar with the green and the purple non-sulfur bacteria, and you'll be pleased to know that I'm not going to talk about them in this class. One photoheterotroph you might know is euglena, which can get its energy needs from light, and assuming that glucose is in the water, it can get its carbon source from glucose. So euglena can act as a photoheterotroph. However, euglena shows you the complexity of life in these four classification systems because if you take away the light, euglena will cannot act as a photoheterotroph because there's no more light around, but it can then switch to a chemoheterotroph using the glucose to get its energy needs and using the glucose to get its carbon source. Let me see if I can remember this. Oh, yeah. If you remove the glucose from the water, but you shine light on the water, euglena can then act as a photoautotroph. And that is, it can get its energy needs from light and can it get its carbon from CO2. So euglena can act as a chemoheterotroph, a photoheterotroph, or a photoautotroph, depending on what environment it is in. Any question about any of that? Um, Let me remind you that the chemosynthetic bacteria can be either a chemoautotroph or a chemoheterotroph, depending on where they get their carbon source. And let me say that besides euglena, we can also say that the chemosynthetic bacteria often act as a chemoautotroph if there's no organic compound for glucose for the chemosynthetic bacteria to get its carbon source, so it'll act as a chemoautotroph. But if you make glucose around, that same species can then act as a chemoheterotroph, and many of the chemosynthetic bacteria will act as a mixotroph, meaning they'll act both as a chemoautotroph, taking CO2 in the cell and also taking glucose into that same cell and then mixing it up where they're working on chemoautotroph and chemoheterotroph at the same time. And some of the chemosynthetic bacteria are that way, where they're mixotrophs and they work as both a chemoautotroph and a chemoheterotroph. And that's why we call it a mixotroph. Any question about any of that? All right. For this nutritional classification of organisms, you must know the classification scheme shown in red. We have all organisms. We can divide them by their energy source. A chemotroph is one that gets its energy needs from a chemical, and a phototroph is one that gets its energy needs from a light source. You can then split the chemotrophs by where they get their carbon source. If they get their carbon from an organic compound, such as glucose, we call it a chemoheterotroph. If they get their carbon source from CO2, we call it a chemoautotroph. If they're a phototroph and they get their carbon from an organic chemical compound such as glucose, we call it a photoheterotroph. If they get their carbon source from CO2, then they are a photoautotroph. You can further divide the photoautotrophs on whether they're oxygenic photoautotrophs or anoxygenic photoautotrophs, meaning do they generate oxygen? And like I said, I won't test you on that, but you could divide the photoautotrophs into oxygenic ones, making oxygen, and then anoxygenic ones, ones that do not make oxygen. Let me remind you that the chemosynthetic bacteria can act as a chemoautotroph or as a chemoheterotroph, depending on where they get their CO2. With the chemoheterotrophs, you can look where the final electron goes. What's the final electron acceptor? And if the final electron acceptor goes to oxygen, then they are engaging in aerobic respiration. But if the final electron acceptor does not go to oxygen, then they are engaging in anaerobic respiration. And you can have two types, fermentation and uh, anaerobic respiration, which we really won't talk about. Well, we will talk about fermentation and we will talk about anaerobic respiration. So I guess it's a good thing that we are mentioning it here. All right. When we're talking about nutrient production, nutrient molecules have energy associated with the electrons that form the chemical bonds of that molecule. In cells, energy is obtained from catabolic reactions that are stored in the chemical bonds of this nutrient molecule. ATP is the energy carrier of cells, and usually when a cell needs energy, it will split off a phosphate or two phosphates from ATP because that third phosphate is a high energy bond. And if you break that bond, you release a lot of energy, which can be used by the cell for its energy needs. And when we break ATP down, that's for catabolic reaction, you break ATP, which has three phosphates, into ADP, which has two phosphate, and then the phosphate's broken off. To make energy in cells, we perform oxidation reduction reactions. Oxidation is the loss of an electron. So molecule A is losing an electron. So we say molecule A becomes oxidized. Molecule B is gaining an electron that comes from molecule A. And so we say molecule B is reduced because it has gained an electron. Oxidation reduction reactions are always coupled. You always have one molecule being oxidized, the other molecule or atom being reduced. And here is an actual presentation where copper plus 1 ion loses an electron, becoming copper 2 plus. Iron 3 plus gains an electron, and it'll change to iron 2 plus. So iron is becoming reduced, and copper is becoming oxidized. An easy way to remember oxidation and reduction is to say Leo the lion goes grr. Leo, lose electron oxidation, grr, gain electron reduction. Any question about any of that? In biological systems, oxidation reduction reactions normally have oxidation having the proton, the H plus, removed with the electron. Remember, in a hydrogen atom, all you have is a H plus ion and an electron minus one. So therefore, in biological systems, biological oxidations are often called dehydrogenations because this molecule becomes oxidized, losing an electron, but it loses the hydrogen at the same time. And then NAD plus, which gains the electron, also gains the hydrogen, becoming NADH. Any question about any of that? So the protons, H plus, are typically also gained with the electron. Biological redox reactions are used in catabolism to extract energy from nutrient molecules. Highly reduced compounds, that's a compound with many H atoms, are degraded or catabolized to highly oxidized compounds. For example, glucose is a highly reduced compound because it has many hydrogens in it, and it'll become oxidized to CO2 and water. The energy, or at least some of that energy being released by glucose, can be trapped to make ATP, and then that energy will be stored in ATP. So highly reduced compounds contain a large amount of potential energy, and if you don't know, obviously, if I were to burn glucose right here, I would have to go several steps back, uh, like if I were to light it on fire by taking a blowtorch to it, because so much energy would be released, I'd have to stand back to avoid the heat. And glucose has so much energy in it, it can actually be used to make a bomb. I don't know what the other ingredients are, but one ingredient of some bombs is glucose. All right, we're going to talk about aerobic respiration in this lesson. Here I'm showing you the outline of aerobic respiration. That is the conversion of glucose in the cell and oxygen into ATP and carbon dioxide and water. In the process, energy is given off, and some of that energy can be stored to make ATP, and some of that energy will be given off as heat. Remember, whenever you convert one form of energy to another, you always lose some of that energy as heat. It's the second law of thermodynamics. The summary reaction for aerobic respiration is glucose, C6H12O6, plus 6 oxygen molecules, plus 38 ADP molecules, and 38 phosphate molecules. This is inorganic phosphate, why it's PI. Make 6 CO2 molecules, 6 water molecules, and 38 ATP molecules. The summary reaction for aerobic respiration. Let's talk briefly about the generation of ATP. Some energy from some redox reactions are used by the cell to make ATP. Animals are lousy at making ATP, and we only have two ways of making ATP, and that's by aerobic respiration or by fermentation. Green plants, which are a little better, can make ATP in a few other reactions, but remember, most catabolic reactions, even in a green plant, cannot generate ATP. The first way that cells can generate ATP is by phosphorylation, the addition of an inorganic phosphate group to ADP. So you have ADP, you add energy, phosphate, you can make ATP. That squiggly line represents a high energy bond. And to get that energy, you can break that bond, and then that releases a lot of energy. The second phosphate also is a high energy bond. And if the cell needs further energy, it could even break off that second phosphate group and use that for its energy needs as well. All right, so the first way of making ATP is by substrate level phosphorylation. Cells have three mechanisms for making ATP. Substrate level phosphorylation, which I kind of showed you here. You have a phosphate group attached to one molecule, and you break it off that molecule and then add it to ADP, making ATP. So you're transferring that phosphate directly to ADP. This actually happens in glycolysis, which we'll talk about later in this lesson. The second way that cells can generate ATP is by respiration. Another name for that is oxidative phosphorylation. This involves the sequential transfer of electrons from an electron donor cell, which is usually glucose, to a final electron acceptor. In aerobic respiration, the final electron acceptor is oxygen. And along the way, the electron is transferred from one molecule to another until at the very end it's transferred to oxygen. In this electron transfer chain, energy is released, and some of that energy can be generated to make ATP. And we'll talk about that in aerobic respiration. Now, note, I've stated that the second way of making ATP is by respiration. This could be aerobic respiration, but it could also be by anaerobic respiration. Any question about any of that? The third way that cells can produce ATP is by photophosphorylation. It occurs only in photosynthetic cells. Light releases electrons from chlorophyll. That light energy is converted into chemical energy via an electron transport chain. We'll talk about that at the end of this lesson. And ATP and NADPH are generated. However, the ATP as well as the NADPH are often used up, meaning broken down, to make sugar. But in photophosphorylation, ATP is made. It's just that it can often be broken down to make sugar. And here you're seeing photosynthetic organisms, why the boys are all coated in mud. Any question about any of that? That's a little bit of a joke there. The plants in the background are photosynthetic.
It's 6:22.
Oh, I've got 620 here. Okay, I was going to stop. Are there any questions? If not, I'll see you in the lab at 6:30. Let's see this. Bye.